Hello everyone, Happy New Year! Welcome to the first MaxWell webinar for 2021. My name is Obien and I will be your host for this webinar. And I'm very happy that we are having already this first webinar here in this month. Next slide, please. So for this webinar, which is actually our episode 6, we have Szilard Sajgo, who is our R&D scientist, who will introduce MaxWell Biosystems and an introduction to our topic, which is spike sorting. And we have our guest speaker, Pierre Yger, from the Vision Institute in Paris, who will discuss about the main presentation. So Pierre is currently a junior PI at the Vision Institute in Paris, and previously he was a postdoc at Kenneth Harris Lab and also in Roman Brett Lab. Currently, he is our user, and he's expert in dealing with the data that Max 1 and Max 2 is giving out in terms of spike sorting. Okay, next slide. So for this webinar, we will have, as I mentioned, an introduction by Szilard and then a presentation by Pierre. And the title of this presentation is Fast and Accurate Spike Sorting for Thousands of Channels. Pierre is a developer of the spike sorting tool called Spiking Circus. And towards the end, we will have the Q&A together with a demo of the Spiking Circus software. And if you would like to use it, please feel free to ask questions. On the right side, you will see our chat. You can type in anytime your questions, and we will be able to answer them at the end of the talk and during the Q&A, and during the demo of the software. And also, we will put in here some links every now and then, depending on what the topic is that is being discussed. For example, linking to Spiking Circus, instructions on how to install it, and also the manual, the documentation on how to use it. All right, so I'll now transfer to Szilard. Szilard, hello.
Hello, and welcome to our webinar, and thank you, Mary, for the nice introduction. So let me just start by introducing to you what we do here at MaxWell. At MaxWell Biosystem, our mission is to provide electrophysiology systems and solutions for advanced scientific discovery and to try to accelerate the novel drug discovery process. Our core expertise is focused on different aspects of MEA technology, including but not limited to hardware and software development. Our customers are mostly academic researchers, contract research organizations, and the biotech and pharmaceutical industries. What is our technology about? So classically, scientists can characterize electrically active cells in two ways, or cells in general. On one side, you have the gold standard, which is electrophysiology using patch clamp glass pipette, and this is used in order to study the electrical currents of neurons. On the other hand, you have the more classical analysis, the imaging analysis, where you can actually characterize the cellular morphology and the subcompartments of the cells. Now, our technology falls somewhere in between of these two techniques. As you can see in the example in the middle, we can not only record the activity of the neuron, but also its morphology. And now you will see the morphology of the neuron reconstructed based on its electrical activity. Now, what is this tool about? So this tool, what we have here at the company, it's an electrophysiology tool that, for instance, comparing to patch clamp, where you record the intracellular electrical signals, here we have tiny sensors that are located outside of the cells, and they record the extracellular electrical activity of neurons. Now, with the application of high-density microelectrode arrays, we have a multiple of these tiny electrodes at really high density. And because of this, we can record from one single neuron in multiple points along its axon, its axonal initial segment, its dendrites, and so on and so on. What are our key advantages of using such a high-resolution MEA? One of the key advantages is that we always have an electrode in the correct positions, thus we can record from every single cell in your culture or your preparation. while in low resolution MEAs, you can miss the cells that are lying in between the electrodes. The other added advantage is that we can record from one neuron in multiple spots, meaning that if you have a more classical system where you record multiple cells on one electrode, you will only see an average signal. And all of these advantages benefit the readouts, where you can do proper network level analysis and also cellular level analysis and also subcellular level analysis of your electrical activities. What are our products? So our products are MaxOne and Max2. MaxOne is the single well MEA format that uses the MaxOne chip, and Max2 is the multi-well mainframe. This is the data acquisition box that has a built-in incubator that can regulate the temperature, humidity, and carbon dioxide concentrations. And for the Max2 mainframe, we are using multi-well HD-MEAs, either in 6-well or 24-well formats. Now, both MaxOne and Max2 use the same chip format, and every single well includes 26,000 platinum black coated microelectrode arrays with a recording area of 2 times 4 millimeters square that can provide very low noise readouts. And also these electrodes can not only record the electrical signal, but it can also stimulate your tissue with electrical signals. Of course, centered to our system that controls these systems is our software. It's called MaxLab Live, and it allows us to visualize and acquire the high quality data, to organize one's experiments, to analyze data and generate summaries, and also to export your data in various formats. It includes different live visualization tools. It can visualize in the format of each individual electrodes. You can see here on the left top panel, each little square is an electrode, and then each red triangle indicates the activity of the cell on that electrode. It can also visualize the channel raster. It can visualize the activity on the electrodes, and it can also visualize a color heat map of the live activity of your tissue, as evident in the video on the right. Now, our software also includes 3 basic assays to get you started in a relatively easy manner using our software. It has activity scan, network scan, and exon tracing. In the activity scan, the software can tell you where your active neurons are on the chip, and you can extract the whole sample activity map, basically label-free. Once you know where your active neurons are on the chip, you can perform a network assay where you can try to answer certain questions about your culture. For instance, whether your neurons are interconnected, do they burst, and whether they are burst in a synchronous manner. You also can use exon tracing to really look at your neurons at subcellular resolution, and you can use exon tracing for this, and you can visualize, are your exons and are your neurons developing correctly? Are the exons of your neurons functional? Are they healthy? How fast is their exon propagation velocity? So all of these you can relatively easily now answer with the exon tracing assay. Now, these 3 assays can be applied to neurons, but of course our system is suitable for many other tissue types, including iPSC-derived neurons, primary neurons, brain slices, 3-dimensional brain organoids, neurons, spheres, retina, but potentially also can be used for cardiomyocytes, muscle cells, or pancreatic cells. Now, unlike in cell culture, where you only have, let's say, 1 or 2 cell types at the most, in certain cases, like acute recordings, like for instance retina or the brain slice, you can be dealing anything from 10 to 100 different cell types. Just a simple notion of the cell types is that these are not only morphologically different, but these are also physiologically different. Now, take the case of the retina. The mouse retina has approximately 30 different ganglion cells. Now, these ganglion cells have spiking activity. and they are modulated by different visual inputs. Now, why would you need to do this process of spike sorting, which Pierre will discuss in more detail later? Now, let's imagine you have a piece of retina placed on your MEA, and you have a moving dot or a moving bar that is moving with different orientations. What you would see in the channel raster, you would see that these cells at certain points fire, they stop firing, they fire, they stop firing, but you cannot determine any temporal properties of individual cells. Now, once you would perform this method called spike sorting, you will be able to assign for each cell what was its temporal properties, for instance, for all three of these directions. You will be able to tell that a particular cell type is direction selectivity for upward motion, downward motion, and so on. So this is the added advantage of doing spike sorting for your tissue. Now, what lies at the heart of this process, to try to explain it in a more simple method? Now imagine that you have the retina, as I mentioned before, and this retina has approximately 400,000 cells per square millimeter. So you have a lot of neurons. To use an analogy on these recordings, imagine you have a room, for instance, here on the left, and it's full of revolutionaries, right? So there's a large chaos, everybody is talking. So all these individuals consider them as retinal ganglion cells, right? And imagine that now you are a spy in the room and you have, let's say, 4 microphones in the room, and you are given the task to reconstruct all the individual discourses. And this is a very classic cocktail party problem. And you are tasked with the following. You need to know how many people are talking in the room. And for this, you need to sort and analyze every person's voice based on the shape of its frequency. And of course, you will encounter several problems. Like some people speak louder, some people speak softer. People that are closer to the microphones will be heard more clearly than people that are farther from the microphones. And of course, you will also record some noise in your microphones. And one of the questions is, how can you potentially improve on your process to be a better spy? Well, one way is to basically add more microphones in the room. And once you can add more microphones, then you can have a microphone closer to each individual. And this could really potentially help you to identify them. Now, however, this also creates a lot of computational challenges for the process called spike sorting, which Pierre will now discuss in more detail in his presentation. And with this, I would like to welcome Pierre.
Hi, everybody. Thanks a lot, César, for the nice introduction and for the talk. So, okay. Yeah, so today I'll talk about the process that Szilard just mentioned, which is spike sorting. How do you turn the extracellular signal recorded by all the channels into spike trains, and how are you are able to disentangle individual units activity? And so the reason we do that is that because I'm working at the Vision Institute, and at the Vision Institute we are really interested in understanding how does the brain encode information along the visual pathway. And the first step for us is the retina. The retina is already still, it's a layered structure where light goes into the photoreceptors, and then the output of the retina is, we know, is given by the retinal ganglion cell. And we can record them by making an ex vivo preparation and putting such a piece of tissue onto a dense MEA. And what we want to obtain to study how is information encoded is we want to obtain a so-called raster plot. On such a plot, every line is a cell, in this particular example, a ganglion cell, and every dot is a spike. So we want to be able to know when every neurons, and as many neurons as possible, because to understand the population activity, the more cells we have, the more data we'll be able to grasp and make models and understand. And we want to get those so-called raster plots. So, and yeah, at the end of the day, we want to link them to the image that were presented and understand the coding properties of the retina or the visual pathways. So if we want to record nowadays from a large neural population, you don't have that many options. You mostly have two options. Either you go for something called high-density electrophysiology, which is like either in vivo you use a dense probe such as neuropixel, or in vitro you can use a dense high-density MEA such as the one developed here by MaxWell, or you can use calcium imaging. This is another technique which is also used a lot, and both techniques will aim at producing those so-called raster plots. So they will both try to provide you the activity of hundreds, if not thousands, of neurons as function of time, if possible, at the individual spike resolution. The problem, however, is that behind those two arrows here that are just very small, there is a very complicated post-processing procedure here. To go from the spike to the extracellular signal to spikes, it's not that simple. And similarly, also for calcium imaging, to go from the fluorescence traces of your cell loaded with calcium into spike is also something complicated. So it is a spike sorting or calcium deconvolution. So today we'll talk about spike sorting. And I want to explain you briefly what's the canonical workflow for spike sorting, or at least what it used to be up to 5 years ago, I would say. So you have your cells lying on top of a dense recording arrays. And in this particular example, it's retinal ganglion cell, but it could be any neurons or any tissue you'd like. What you record by what the channels are recording are the extracellular potential. So it's the sum of the extracellular, so it's the sum of the spikes plus some what we call the local field potential. Because we are not interested in the local field potential, we really want to focus on the spike. Most of the algorithm, what they'll do is that they will pre-process the data by filtering them with a high or bandpass filter, getting rid of the slow fluctuation, and keep only those high frequency events that are the spike. Eventually the data will be weighted, but I won't explain here this step. So having done so you get your traces, you can detect the spikes, or at least the event at which you think a spike is occurring, because you can use a threshold detector, and when you have a threshold crossing, you can highlight this particular moment of your recording where there is an event. Having done so, again, you can collect those particular events, align them as function of the time of the peak detection, and you get a collection of snippets, called them snippets, or waveform, spatio-temporal waveform, because they are collected across several channels. And the goal of spike sorting is to take those waveforms, and I won't explain how, but project them with a dimensionality reduction technique into a lower dimensional space. Quite often we can use PCA, but it could be singular value decomposition or wavelet transform. It doesn't really matter, but the goal is to project those waveform, those snippets, into a low dimension space where we can cluster, meaning we can run a clustering algorithm that will assign labels to the points, and that will decide, in a sense, that the dots on the left are the purple or the pink, and they belong to to a particular template. So what I will call a template is the spatio-temporal motif elicited on your recording channel when a given neuron is firing an action potential. And having done so, then you have all the points on the left or on the right, and you can assign labels and say that you found the spike trains for the unit 1, putative unit 1 or putative unit 2 that have different waveforms or templates. Actually, this pipeline, when I say I used to be working up to 5 years ago, works for at most tens of independent channels, which is clearly not the case here when you have a dense array. And it's even worse, it's also not really accurate if we want to study fine correlation, and I will explain why in a minute, which is also something problematic, especially if, like in our case, we want to work on raster plots and correlation properties of the information. So we need to address this issue. How do we do that, actually? And what's wrong with those 2 points? So the first, why is it not working when you have a lot of channels? It's because clustering algorithms that are used in the assignment of the labels do not scale well with the number of data points. So if you have longer recordings and a lot of spikes, which is the case nowadays with dense arrays, all the clustering algorithms that you'll find are scaling badly, meaning they will take all the memory and take a lot of time. So there is a computational bottleneck here that you can't solve. And the other problem, which is related to the correlation, is imagine the following situation. You have a cell, neuron A, which when it's firing a spike leads to elicit this blue pattern, the blue template. And you have neuron B on the right that elicit a different pattern. The two templates are similar, but not exactly the same. The summa of those two cells are likely to be next or close to the same channels, but the shapes of those templates are a bit different because of the morphologies, the axon, the dendrites. So we can distinguish them, but they look alike. When cell A is firing alone, then a spike of A will fall into this blue cloud, and it will be easily detected by a clustering-based algorithm. Same for B. A spike of B alone will fall in the red cloud and no problem. The problem arises when A and B are firing together simultaneously and with a collision. And when it's happening, in green, you have the trajectory of all the when you take templates A and B and you superimpose them with all the possible lags. And so you start from here when A is alone, then you start adding B, you go there, you go into B alone, and you come back. And you see that all the points in the middle, those are outliers for a clustering-based algorithm. They will be hard to label, and even you as a human, if you had to detour those clouds, you will ignore those spikes. But those are real spikes. Those are superimposed waveform or collision. And it's a pity or it's a shame not to use them, especially because they are now becoming more striking with dense array. So yeah, why is it getting worse, especially with those arrays? So we have an increasing number of electrodes. So as you may have followed in the literature, so in the 60s we're recording from one channel, but so the tetrode, 4 channels in vivo, and the NeuroPixel were released in 2017. So it's 400 channel almost in vivo, and the trends keeps increasing. And same in vitro. In vitro, the MaxWell array or those new CMOS-based chips have thousands of channels, and nothing really prevents them to keep, for the density of the channel to keep increasing. So we have denser and denser probes, which is good on one side, because with dense arrays, when a neuron is firing, the spike of this particular cell is seen on many channels. So the template that will record on the channels is really unique and will allow us to identify this cell and label those spikes. On the drawbacks is that channels are also seeing many cells because they are getting denser and denser. So we have a lot of collision. And it's rare that a neuron is firing alone, actually. There are always spikes nearby, and so we always end up having collisions. So the overlaps that I just mentioned before are now the rules with the dense arrays, so we need to find a way to solve them. And yeah, and it's getting worse because, as I said before, the future is coming in a sense. We can think about, so NeuroPixel 2, for example, in vivo is on the way with several chunks or stacked NeuroPixel. We can think about 3D probes, so the scalability of the algorithm needs to be an issue. So we need to have ways to deal with that. So yeah, the solution that was taken by the field is to follow what's called a template matching approach. And so spike sorting now is done slightly differently compared to what it used to be done, but it's done using an approach called template matching. So the signal S, your extracellular signal of all your channels, we try to explain it or to as a linear sum of motifs or templates, or yeah, the W with so every time you have a spike, you have the motif triggered by the spike onto the MEA, which is eventually weighted by a scaling factor. Could be 1 or it could be something else, plus some noise. And so this is just the mathematical formulation of the problem. The good point about stating the problem in such a way is that we are facing an optimization problem. We need to find, okay, the spike times, the templates, and the amplitude factors. But this is something we can do mathematically. And by doing so, we could also avoid clustering from all the spikes and get a dictionary of templates only from a subset of the spikes. And with this linear approximation, which holds and it's valid based on what we know from the properties of the extracellular media, we can solve the problem of the overlapping spikes. So the pipeline that I just showed you before is now slightly modified in a sense, but the picture here is the same. But we detect from a subset, we detect a subset of spikes. We align only a portion of them, a subset. The clustering is run on a subset, and so we obtain a dictionary of templates only by looking at a fraction of your data. We build this dictionary of templates, and having this dictionary, we ask ourselves, okay, how can we reconstruct the signal as a linear sum of those motifs? And it's only during this template matching procedure that we assign the labels and we get the spike trains. So there is a whole diversity of software nowadays. You may have heard about some of them, like Kilosort, Yas, Spiking Circus, which is the one we are developing and that I will briefly highlight today. The one in reds are template matching based software, meaning they follow this approach of trying to reconstruct the signal as a linear sum of motifs. They are all somehow unified within the Spike Forest Spike Interface Initiative. And Spiking Circus, yeah, is the one we are developing. It has been published in eLife in 2018, and it was based on some pioneering work in 2012. And it has all the key ingredients I just highlighted to you. It's clustering from a subset of the spike, and in order to obtain a dictionary of the motifs quickly and robustly. Once this dictionary of motif is obtained, then it's fitted onto the data and we get the spike times. It's scaling nicely for thousands of channels. That was our purpose. And it was extensively validated, and I will explain you in a minute how. So the software, because we wanted it to scale for thousands of channels, has an optimized trade-off between speed and memory consumption. It has been tested, and I will show you in a demo after with MaxWell Biosystems software, but some others also. It has an it's meant to optimize the memory footprint because, yeah, you have a large number of recorded channels, so you can have huge templates. So it's meant to be launched on a single machine with quite a decent amount of RAM, but still. And you can have thousands of templates with adaptive spatial support. So for us, it was also something important because when you record some particular tissue, like retina, for example, the templates can be arbitrarily large because, for example, when a spike is propagated along the axon, it can be seen up to hundreds of microns away from the soma, but it's still the same event, and you would like to be able to capture it as a single motif or template. So this is something important. But it's more like a technical details. Internally in the software, everything is handled as sparse data structure and doesn't really use the GPU at the moment. So when I said it has been extensively validated, it has been mostly validated in two ways. First, with real ground truth recordings. So in our case, what we did was that we patched juxtacellularly a cell in the retina while simultaneously recording with the dense array the extracellular activity. And you know that you know the spikes of this guy because you've patched it, and you have the extracellular recordings. And you can ask yourself the question, okay, if I'm spike sorting the data, am I able to recover the spikes of this particular cell? So the question is easily phrased. The answer is more complicated, but the outcome is yes. And I would say that if your cell is nicely visible from your MEA, meaning not too far and close enough, then you can have error rates at about few percent, which is really good compared to techniques such as calcium imaging. If you are interested into more benchmarks, actually, there is the Spike Forest initiative that I mentioned before. There was a paper published in eLife, again in 2020, where the goal is to benchmark all modern spike sorting softwares on various datasets. Could be real ground truth experiment, as the one as I just highlight, or hybrid datasets or synthetic datasets. And all those Spike softwares are run with default parameters on plenty of datasets. And on this website, if you have a look to the paper, you will have the results or the performances. Because, so I will not be able to tell you that there is one software that will be the best one, regardless of the tissue type or anesthesia, whatever. So it's still something which is under investigation. So what kind of software you need to use depending on the particular tissue or stuff. What's sure is that it's working nicely in our case with the MaxWell device. So here, for example, you'll have a view in Spiking Circus of the layout of the recorded channels. So those devices have a lot of channels. You can select a subset to record from. Here it's 730 that are recorded. So we highlighted the subset, just 16 of them. So you have the raw traces. And Spiking Circus will provide you, after the completion, a reconstruction in black here, which is superimposed, of templates and spike times, such that if you want, you can, because you have the templates, the spike times, and the amplitude, you can reconstruct your signal and see that you have a nice fit here. So, yeah, to conclude, in a sense, the outputs of the spike sorting pipeline, this is what Szilard was mentioning before, is that you have one template for every putative neuron, meaning you have the templates as the output. So the spatial temporal motifs elicited on your recording channels when a given cell's fire. Then you have the spike times here. I will come back in a minute to the explanation of those panels in a demo where you can ask a question if you want. But so you have, for every cell, you have its spike time. So when do you have a spike? And you have what we call the amplitude. So the amplitude factor I mentioned before, at which the template is fitted onto the data. As I said, it should be 1, but it can vary a bit, and it's important to look at it because it's a way to assess if your cell is drifting. Because I will not go into the details here, but you can have drift if you have a long recording or non-stationary recording. Your cell may slightly move a bit, which will lead to a change of its waveform, and you need to track it. And those algorithms are still somehow what we call semi-automated, meaning you still have some manual curation at the end, because the code will output you template, spike time, and amplitude, but human will still need to rank the template based on the quality, maybe, and eventually correct for over-clustering. This is something also we can discuss in the hands-on session, but the problem you can easily guess arise if you have a cell, for example, drifting during your experiment. You'll have one template for the beginning of your experiment, or also maybe if you add drug, the drug will affect the waveform shape, such that at some point the cell will have a different waveform. And the code will find 2 templates, and it's only you as a human that will need to understand or make the junction. So this is something which is still also not automated yet. And if you have artifacts also in your data, then they need to be thrown away and by an expert eye. So there is still some part here of manual assessment. So to conclude, in a sense, on the software, the strengths and the challenges is that it's widely used. Actually, we have a lot of users in many labs and citation already. So it's, as I said, it's validated, and it's, I would say, easy and friendly to use. It's there is a linear speed up according to the number of cores you're using. So if you're not satisfied on a single machine, then you can stack several, and you get a speed up. There's good performances. It's user friendly, or I mean, as user friendly as we can do while developing it, meaning it can handle file formats, stream. If you have several chunks of experiment that are recorded, you can concatenate them virtually and process them as a whole. And it also can be adapted for real online spike sorting. The challenges that are still ahead for us as developers is that, as I said, a more robust handling of the drifts, something that will automatically detect when a cell is drifting and correct for that. Maybe think about better default parameters, especially for NeuroPixel, HD-MEA, or spatial tissue. As I said, currently, we have default params that we don't really tweak that much because the parameter space is huge. And yeah. and speed improvement are always welcome, of course. So just to highlight, in our case, because the software was also meant to work for that, but we have a beta version of it working in real time, meaning that's what we want to do at the end. Close the loop and get the extracellular recordings and the spike train on the fly, such that we can modify the stim accordingly and close the loop. That would be really great to study the population responses to stim dynamically. So this is something that we are actively working on, actually. And yeah, and I would like to thank my collaborators, Olivier, Mar, Baptiste Lefebvre, Andreas Sormento, two PhD students. And I would encourage you to have a look to the website and the documentation, which is, I hope, yeah, sufficient. And otherwise, I can make a hands-on session. So I will share my screen. And I don't know, feel free to ask questions, maybe to guide me regarding what to display or show, because, okay, I'll start sharing the screen.
Thank you so much, Pierre. It was a really very nice presentation showing us how spike sorting works, also introducing spiking circus, and we are excited to see now the demo. While you are preparing it, I'm just getting some questions already now.
Okay.
Yes. So one question that we have here is, during the course of an experiment, is spiking circus determining the templates multiple times? If yes, how frequently? I guess this works with an online approach.
Yeah, well, so during the offline approach, sorry, I will come back to the slide here. It's, for the offline version, the templates is only fixed, I mean, is fixed during the course of the experiment. Okay, so there is one template, and the code allows the template to be distorted a bit during the along the course of your experiment. So, I mean, if you have a drift which is not too strong, meaning if it's just an amplitude scaling or a slight distortion, then the code will compensate for that, and it will be reflected in the amplitude factors that will change, but you will still track your neuron nicely. If you, for example, have a strong pharmacological modification at some point, then, and which is really messing around with all the shape of the templates and the firing properties of your cells, such that all the waveforms are shuffled in a sense, then there are no mystery there. You will have templates active up to the time where you put the drug, and then you'll have a new set of templates somehow active. And currently, the code will not be able to say that cell 1 is the same as cell 2 after the time of the application of the drug. So you'll have twice the number of templates in a sense regarding to the number of cells. But to be honest, even human will have a hard time to make the junction here. So it's complicated. So regarding drifts, this is really an open issue, and it depends.
All right. Yes, and I have kind of a follow-up question on drifts. Are there any kind of plans on how to improve the drift handling of the current implementation of spiking circus?
Yes, so actually, when you have a tissue and the probe and some pressure, you can have an abrupt changes, and it's much more complicated. So here, yes, we have ideas about how to compensate for that, and this issue, and on an image-to-image basis, the frame are slightly moving, and you need to compensate for that. So you have what's called rigid, or you have a registration procedure that will realign all the frame onto a reference frame. And currently, the trends in spike sorting, so it's true in kilosort or for YAS, or also for us, is to try to register also the data during the pre-processing step. While filtering, we register them also, such that we will compensate for a large portion of the drifts even before the sorting, I would say, just because of that. So yes, but to be honest, yeah, in vitro with those dense arrays, if you record for up to hours, drifts are not that problematic.
Yeah, I agree. Yes. All right. Okay. So Yes, maybe we can proceed to the demo of spiking circus, and I would like to invite Szilard again back to the screen so that we can
Hello.
have a discussion on what would be nice to demo here for our audience. So maybe if you can show Pierre just what type of software
Yeah. Okay, okay, let's oops. I have my website. Oops. So as I said, okay, let's start with the beginning. So this is the documentation website, okay, of Spiking Circus. So you'll find all the details regarding the algorithm, how to install it. So it should be easy to install. It's cross-platform, meaning it's working on Linux, Mac, or Windows. It's a Python package, so the installation should be straightforward. And you have all the details regarding the parameters and how to use it. As I said, so the software works as a black box, meaning the goal for us was to target non-advanced users, such as you just want to load the datasets, launch the sorting, and get the results. However, you can still have access to all the parameters and tweak them if you want. But yeah, it's up to you, I would say. It's really if you're not satisfied with the default results and if you think your data have something really special. So you can launch the software in two ways, either with a command line if you're, I don't know, yeah, inexperienced Linux or Mac user and you work with command line, or you can have a graphical user interface, a very small GUI, which does nothing really fancy, but allows you to launch the command line for you. So actually, Szilard gave me some data sets from Retina, mice Retina here in response to flashes. So I can just, for example, if I'm opening one of them here, so yeah, in the GUI, you'll see all the parameters. This is the parameter file. So the code will build the parameter file. Don't be afraid of the number of parameters. As I said, we made the choice to expose all of them, but the goal is to not touch any of them. You just need to say that for the file format, you have a MaxWell file format. The code will not detect it automatically because you need to actively say it. And having said so, it will automatically then get the metadata out of the file and be able to do something out of it. So then the sorting have already been launched here. So I will just immediately go to the results of the code. And actually, yeah, because I was preparing the presentation, oops. I launched, so we have two ways to visualize the results. So either with a Python GUI, which is not developed by us, but which is developed by an engineer called Cyril Rossan in Kenneth Harris Lab, working in the International Brain Consortium. So this is a GUI where actually most of the spike sorting software aims to plug themselves, KiloSort, we, and ourselves. And in such a GUI, what you can visualize is all the templates here that have been found by the software on this particular data set. You can sort them as function of the number of spikes or amplitude of the peaks and so on and so forth. And here, for example, this is what you'll see by selecting one of them. So you see that so this is the shape of the template. So meaning this is the spatial temporal motif found by the software on your MEA. So you can, if you want, show the label of your probe or not. And this is the plot that I mentioned before. So every point is a spike. And this is the amplitude over time. So it's around 1, but it's not always 1. It's a Gaussian distribution, so you don't have any drift. It's pretty stable. And you see, so here in this data set, so the tissue is responding to flashes of light, if I'm correct. And you see nicely that you have cells that indeed respond to onset or offsets of those flashes. So for example, here, the spikes, the cell responds. So maybe we'll have some other cells that, so some cells, as you can see, can have very, very broad templates that covers a huge span of the electrodes. Some are more confined and compact. But in all those cases, here you see this is the firing rate of the cell as function of time. This is the so-called interspike interval view. And this is something important in the spike sorting community because most of the people use such a view as a quality criteria for the spike sorting because you know, or you can also look at it in this plot, which is the autocorrelogram view, meaning it's the interspike interval view means that it's the distribution of the time interval between two consecutive spikes. And if you have a real neuron, because you know that the real neuron cannot physically fire twice in less than the refractory period of dilation, you expect this distribution to have a dip here at 0 and to have this gamma shape where it's really going up and down. So this is a sign that this is a cell in a sense. And you see, so here, for example, this is a cell which is active before the flashes, then you see it at those flashes. And it can reflect nicely in the PSH. And in such a GUI, the GUI is pretty nice because so you can select a subset of them, you have all the templates, you can look at, so then this view change, and now you look at the cross correlation between all those pairs. Cross correlation means it's when you have a spike of cell 1 and cell 2, what's the distribution of the interval? And again, as I said, the major question here that people are asking when you do spike sorting. So as a human, you need to assess the quality of those templates. So Spiking Circus, as I said, we are working to provide you a real fully automated sorting, but it's not yet the case. But Spiking Circus outputs you what we call a purity number. So it's a number that should be 1 if we are really confident that this is a cell and you have all its spikes, and it should degrade and be kind of not as good if we are less sure about the results. So if it's close to 0, then we can't guarantee that this cell will not also be a bit corrupted by noise or something like that. Because what you really need to understand is that spike sorting, so Szilard mentioned it before, but it's a blind source separation problem, but it's mathematically it's a nil-defined problem because you will always nicely record the cells that are close to your electrode, because when they will fire an action potential, they will elicit a waveform which is strong enough to be detected. But you will always have cells that are further away from the electrode that will be captured by your array, but not precisely enough such that you will not be sure that they are nicely isolated. They will be fading away in the noise, so to say. So this is something, for example, where the autochoreogram doesn't really look like it has a clear dip here. The template is only defined on 2 channels, so it's likely to be it's a pattern visible in the signal, but I can't guarantee that it's coming from only a single neuron. It can be a mixture of cells far away. So yeah, that's about it. And regarding the drifts, so those GUIs, if you can always ask those GUIs, they are meant to help you or to guide you for doing the putative merges, so to say, because as I said, the only problem that you can have in spike sorting is over clustering. So if you have a drift, if you have a cell that has been, because it's moving during your experiment or because you had drag, that has been splitted in 2 templates. So you need to kind of be sure that your cell is not splitted into 2. So each time you have a cell here, you can ask, what's the closest template from my particular neuron, and the GUI will suggest you the best match. So actually here Spiking Circus is already cleaning a lot the data, so you should not have on this particular recording, which is pretty short and stationary, there are no drifts, so no merges should be done. But that's it. So any question here regarding the GUI or?
Any questions from our audience? Szilard, do you have something you would like to emphasize or?
Yes, so in very basic terms, when you look at the cross-choreogram, for non-expert users, the way you would define whether 2 cells need to be merged or need to be left split, you just need to look at the center point, the dip, as Pierre said, and you just look at the right side and the left side of the autochoreogram. And now, if these 2 sides are significantly different, then most likely what you have is you have 2 different cells. If they're similar in shape, then most likely you have units that need to be merged. But this is still something, a process, as Pierre said, that you as a human have to define. Of course, all sorts of post-processing of the data and filtering is possible, but you have to define the rules of these post-processing steps.
Yeah, and as I said, okay, before, as I said, spike sorting, when so here in such a small data set, to be honest, we have all, I would say the results can be trusted almost without any curation. But it's up to you, depending on your scientific question, that need to set a quality threshold regarding the cell you want to keep for your analysis. Because if your scientific question relies on very fine correlation or question about coding properties and so on and so forth, you really want to keep only the cells that have very good templates, high quality to noise ratio, and that you can, for example, if you just based on purity said that, oh, everything that's below a certain purity value, I don't want to keep it because I'm not sure. If, on the other hand, your scientific question is more about macroscopic quantities such as firing rate and activity of your tissue, then you can have a criteria which is more loose and say, I don't care because I know that all those stuffs are spikes. I just accept that for some of them, I'm not sure they are really assigned to the good cell, or that maybe some cells are mixtures. We call that multi-unit activity, MUA, because we can't guarantee that this is a single cell. So this is where the human expertise is somehow needed. And if you're not familiar, or if you don't like, so the Python GUI is cool, and save everything as Python arrays that can nicely be plugged into a pipeline after analysis pipeline. But we also have similarly a MATLAB GUI that will basically show you the same stuff. So you just need to launch the command here, or again, you can here you can select Fire MATLAB and it will launch the GUI. I'm using the command line because it's easier for me, but so, and yeah, and the MATLAB GUI will show you the same stuff. So you have the layout of all your channel here and the templates, and you can go through all your templates and see and assess nicely that here you have the, and you see this is likely to be a cell, and as Szilard mentioned before, or in the movie you showed before, maybe you have the axon here or something that goes, and you can figure it out because you see you have the pattern, and you can also look at the peak and see that it's a traveling waves actually going. And this cell is active here a lot, just during the flashes of light. So it's not active before, it's only active there. And you have a very nice ISI. And the other quality metric, which is used to assess the quality of the sorting, at the end, it's what we call the refractory perambulation. It's the number of spikes that are too close in time such that we think it's an error, and usually, so here it's 0, but you see that for some units, yeah, for example, I don't know, I could go back to the first one that was bad, but so this, you have a lot of green and yellow dots. Those are refractory perambulation, meaning here we have 22% of the spikes that are violating the refractory pairs. So we know that this is not a single cell. No way it can be. So either it's noise, or it's multi-unit activity, because it's still something that's visible in the signal, but it's just this small pattern here, and that we see regularly. So actually, yeah. And so that's about it. And like, yeah, question?
Yes. Thanks a lot, Pierre. Now we have several questions actually about the spiking circuit itself, and also how to use it. So the first thing that we have here is if we are doing electrical stimulation and we are looking for the response of the cells to this electrical stimulation, so how, for example, does the template look like, considering that there is a possibility that there is an artifact caused by the electrical stimulation?
Yeah, sure. So actually, spiking circuits as a built-in mechanism. So if you have a stimulation, which is always the same, for example, a flash of light or on the tissue, then you can give spiking circuits the spike times at which the stim is occurring. and the time window during which you think there is an artifact or recovery of the tissue or something like that, because it could be a piezoelectric effect or something. And then what spiking circuits will do during the filtering procedure or the pre-processing, it will look at all the portions of the code at all those simulation time, and compute the median artifact, so and subtract it from the signal, such that we will only focus on what's not on the trial on an artifact to artifact basis reproducible. That's being said, if you really have a stim that's triggering spikes and that are always the same time lock to your stim on top of your artifact, then they will be subtracted too. I mean, there is no way the spiking circuits can distinguish them. I mean, those time locked spikes in response to the stim with regard to the stim itself. I'm not sure I'm clear, but so actually in the doc, there is something here written somewhere dealing with stimulation artifacts. So you provide a text file with the t-start, t-stop, or just the certain, and you can have different stim type that lasts different duration, and you flag them. And spiking circuits will automatically just identify the median artifact, subtract it, such that you will only focus on what's viable. That's I'm not sure it's answering fully the question, is it? Yeah.
Yes, it does. Thank you so much. We have another question, another topic. So now here I'm just reading directly the question. So how the scaled form of the template is normalized in the matching method?
Yes. So the templates are normalized because when we do the template matching, so mathematically this template matching procedure works with templates that are all of equal norm, so norm 1. However, at the end, they are unnormalized. Oh, I will mask that. And you can easily turn them back into the real world of your physical units and have amplitudes in microvolt and something like that. So internally, they are normalized because it has to be for the fitting procedure. But at the end here, you see the templates, they have different norms, meaning they have this is a small one. This is a larger one. They all have the amplitude, which is actually, this is really the motif you see on the raw data, on your filtered data, when the neuron is firing. So it can be directly put into microvolt if you want.
Mm-hmm. Okay. Yes, and now you're discussing also the motifs, right? Or I think for us we call them footprints. And sometimes for the same cell, you see kind of different shapes of the signal. Like there are those that are positive spikes, there are those that are negative spikes. So in terms of spike detection, now for spiking circuits, is it able to determine kind of the different shape of the action potentials at the soma or at the axis?
So okay, so spiking circuits is, I would say, so what spiking circuit does is that it's only it's detecting motifs that are crossing a threshold. By default, this is a negative threshold crossing, because most of the spikes in vivo or in vitro, when you look at the electrode the closest to the soma, because of the properties of the extracellular medium, this is likely to be where the soma is, and it's a negative crossing. However, so that's being said, so we flag those times, but the motif here have this is the the full motifs have different shape on all the other channels. The shape depends on the morphology, whereas the actual initial segment, the soma, the arbor, the dendrite, and that's what are making the template unique. If you want spiking circuits to also detect positive peaks, because I don't know, it has been asked by some users, because it seems that in vivo, in some particular structure, the spikes are not that are biphasic or triphasic with a positive peaks, then you can ask spiking circuits to detect both positive and negative peaks, and you'll have templates that are then combined, so it's the same. But I mean, usually, yes, take the shape of the template to guess where the soma is. So you have also some other algorithm that will try to infer the opputative position for the soma of your neuron based on the templates.
Mm-hmm.
So, and also then after, if you want to go into that, people will usually use the shape of the template to maybe make some claim regarding putative excitatory or inhibitory cells, because we know that inhibitory neurons tend to be more fast spikers, so that they have a higher firing rate, and the template is sharper and narrower. This at least has been observed in vivo. I mean, I'm less familiar with that in vitro, and also in the retina, we don't have this issue because we don't have excitatory or inhibitory cells, in the sense we only have the ganglion cell. But so you can then classify based on some template properties,
Mm-hmm.
peak to peak amplitude or spike, spike width, duration, and so on and so forth, putative cell types. This is what's done in vivo, at least by people, to assess excitatory and inhibitory, at least subgroups.
Yeah. I mean, there are many things that can be evaluated to classify different cell types. And actually, now that you have a rich kind of data set, not only kind of spike times, but also these motifs, yeah, it allows you to really kind of have a more classification data set classified. So I have now another question here. So I think these motifs across multiple days, for example, in cell cultures, they may either stay the same or may change, or there may be a little bit of differences across multiple days. Do you have any recommendation if, for example, someone would like to track the same cell across multiple days and find out if this is really the same cell that is being sorted across multiple days?
Yeah, that's so tracking the same cell over multiple days. So I don't have any experience regarding to that. I just know that, so okay, if the tissue is not growing and if the cell is more or less in place, then I guess you'll be able to find it over days and it shouldn't really be a big deal. If the cell is moving, the tissue is growing, and the waveform is drastically changed, again, you have this issue of tracking, which will be hard. I mean, the software, I mean, okay, first of all, let me say what you should do with spiking circus, and that's what is written in the doc and advocated to all the users. If you have several recordings of the same tissue over multiple recording session, let's say you record 10 minutes, then you wait, you do something, you record another 10 minutes, or maybe a day, and you record again. What you should not do is spike sort the first recording and then the second one and the third one. Why? Because you'll end up with a bunch of templates and unit for the first recordings and for the second. The clustering algorithm is not like it will never be the same. So everything will be shuffled, and it will be hard for you to make the juncture and say that cell 1 in the first recording is equal to cell 37 in the second one. So what we need to do, if you have such a situation, is to process what we call stream of data. You need to virtually concatenate all your file together, and spiking circus can do that simply. You just need to put all the files in the same folders and say that you need to process all the files. And as you can see, when you launch, it will say multi-file, 19 files have been found, and it will virtually concatenate all the files, and the template will be tried to be applied continuously across all those files, such that tracking the same units is much easier.
Mm-hmm.
So this is what you need to do. That being said, this assumes that the non-stationarity between files is not too drastic. So this is why I said before that if you really have a growing tissue and if you wait a week to record, then no doubt that it will be hard to track the cells. But if you record regularly and concatenate everything virtually, it should work. Just also a point to highlight the importance of spike sorting with respect to what has been said before by Szilard. So if you just, for example, some templates, I don't know, like not this one, maybe, I don't know, don't have a good example here. It's always when you search for a good example that you don't have it. But you see that some templates have a big amplitude on many channels and are crossing on many channels. So if you just naively take all your recording and detect the threshold crossing, you will end up overestimating the firing rate, because, for example, here it's only one spike. It's not a spike there, 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 there. So the importance of spike sorting is that your channels are not independent, and if you don't want to count multiple times the same event by processing the channels individually and just saying, oh, I'm interested in the global firing rate, I will count the threshold crossing on all my channels, and you will end up drastically overestimating the firing rate, especially with those dense arrays.
Mm-hmm.
So the channel rasters that were showed by Szilard before and that you have are interesting to know where the tissue is active, not active. But if you want to go into fine measure of what's the firing rate, you end up, you need to do spike sorting somehow to avoid multiple countings of the same events.
Mm-hmm. Yep. Okay, I think we now have just our last question, and then we will end the webinar. So as a last question, in acute recordings in vitro, we have perfusion running. And I guess depending on the placement of the tissue, some are asking if there could be a possibility of a drift caused by, the like perfusion system or by changing media in the shape, for example, of the action potential or in the position of the cell. So do you have any feedback on this, maybe on the retina?
Well, we also have a perfusion and we don't see anything, but so I'm not that concerned, but I don't know. I mean, yeah, if people have some particular data or where they clearly see, I mean, if you should see that such a drift or change in the raw data themselves. If you don't see that here, then I doubt it will affect spike sorting afterwards. So, I mean, we never had to deal with this issue anyway. But
Alright.
So mostly these drifts, they're an issue for in vivo.
In vivo, exactly.
When you have a probe in the brain and the animal.
Well, in vivo, but also in vitro. I'm just saying that because somehow in a culture, I mean, okay, in the slice or in the retina, the cells are dying also a bit. I mean, it's just that you need to face the truth such that cells are slightly dying.
Yes.
So they are changing the waveform are affected slowly. And also just if you have, for example, optogenetics team or some other stuff like that, if you start messing around with some channelodopsin channels or some, as I said, pharmacology, then you are changing the waveforms because the way you make cells fire is not physiological. So yeah, it's different. I'm just saying that. So this is something that needs to be taken into account.
All right. So thank you very much, Pierre and Szilard, and thank you very much to all of our viewers of this webinar. I hope you enjoyed the webinar and also learned a lot about spike sorting and how to use spiking circuits. So again, here in the link already in the chat, we have a link where you can download the software and also where you can access documentation and that Pierre was showing us earlier. And if you have any questions, you can send us directly your questions, or you can also contact directly Pierre through the spiking circuits documentation, I guess. So here we have our contact information. You can email us. You can schedule a call directly with us, and you can also visit our website for more information about our system and some applications. All right, thank you very much, everyone, and have a nice evening or morning wherever you are. And see you again in our next webinar. Thank you.
Thank you. Goodbye.
Bye.
All right. Thanks a lot.
Thank you very much.
Bye.